Humanity is currently trapped on a digital treadmill. Every day, we create more data than our technology can reliably hold. This forces us into a brutal save versus discard survival choice for our collective knowledge. If we do not find a new medium, we will be forced to delete the very data that fuels medical breakthroughs and artificial intelligence. This is not just a storage problem, it is a crisis of human memory. Our current tools, like hard disk drives and magnetic tape, are hitting a wall. The rate of density growth for these media is stalling. Even worse, these devices expire in years, not decades. This creates a wasteful technology refresh cycle. In many large archives, workers must start moving data to new hardware before the previous move is even finished. We are essentially rewriting our history just to keep it from fading away. DNA data storage offers an exit from this cycle as the ultimate archive. It provides incredible information density, packing more bits into a cubic millimeter than any electronic device. Because DNA is the blueprint of life, it is a universal format. It will never become an obsolete floppy disk as long as humans are biological. With zero power at rest, a DNA archive is a permanent, sustainable library that can store our legacy for centuries. To bridge the gap between silicon and biology, we first need to translate our digital thoughts into the language of life. DNA storage is at its core a translation project. Computers speak in zeros and ones, while biology uses 4 chemical bases: A, C, G, and T. The bridge between these worlds is a software layer called a codec. This tool maps digital files into biological sequences with total precision. Mapping this code requires more than a simple swap. Traditional storage uses Hamming distance to find errors, but biology is more complex. We must use Levenshtein distance, often called the edit distance. This allows the computer to account for biological typos like insertions or deletions. To keep the biological printer from jamming, we use ternary representation or transitions between homopolymers. These methods ensure we don't repeat the same letter too many times, which reduces errors during the writing process. To guarantee the data survives, we use concatenated error correction. This is a stack of safety nets. An inner code, like Reed-Solomon, fixes small local typos, while an outer code, like fountain codes, rebuilds the data if entire strands are lost. We also apply DNA sequence constraints to avoid sequences that are hard to build or violate biosecurity rules. Once the digital plan is hardened by these safety nets, we move from the screen to the physical printing of the molecules. The most vital step is synthesis. This is the write operation that turns digital code into physical matter. It is a biological printing press where atoms are assembled into the famous double helix. We have 2 main ways to print this code. First, base-by-base synthesis. Also known as de novo synthesis, this builds a custom strand 1 rung at a time from the ground up. Second, DNA assembly. This is a building block approach. We take pre-made segments of DNA and snap them together like Lego blocks to create longer, more complex sequences. The chemistry of writing is also evolving. The old way, chemical synthesis, relies on harsh organic solvents. The new strategic frontier is enzymatic synthesis. This green method uses water-based reactions and natural enzymes. It is better for the environment and allows us to write longer, more accurate strands. We are even exploring structure-based encoding using DNA origami. In this method, we use tiny staples to fold a long scaffold of DNA into 3D shapes. This allows us to read data using advanced microscopy called DNA paint, looking at the shape of the molecule rather than just the sequence of letters. Once printed, these molecules must be locked away in a vault that can withstand the test of time. A hard drive might last 5 years, but DNA can last millions. We know this is possible because scientists have recovered the genetic code of animals from fossils over 2 million years old. This durability completely changes the concept of archiving. It moves us from temporary storage to permanent memory. To keep this data alive, we must defeat DNA's primary enemies: air, light, and, most importantly, water. Water is the most common catalyst for decay. By using containment systems that dehydrate the DNA, we essentially freeze the data in time. Once dry and sealed in a vacuum, a global library can sit at room temperature for centuries without using a single watt of electricity. Finding a file in a pool of billions of strands is made possible by object tagging. Think of this as the library's Dewey Decimal System. We add tiny molecular handles to the ends of the DNA. This allows for random access, where we can pull one specific document out of a massive archive without having to read everything else. The final journey of the data is the transition back from chemistry to the computer screen. The read operation is called sequencing. A biological scanner looks at the chemical rungs of the DNA and turns them back into digital code. Two technologies lead this field. One, sequencing by synthesis, the high-accuracy gold standard that reads DNA as it is being rebuilt. Two, nanopore sequencing, a portable real-time reader that pulls a DNA strand through a tiny hole and identifies the letters by their electrical signal. Because biology is messy, we don't just trust a single read. We use a process of algorithmic voting known as clustering. Since synthesis creates many copies of the same strand, the computer looks at the whole group. Even if every single copy has a small, unique error, the computer can find the truth by looking at the consensus. This mathematical voting ensures the file you get back is a perfect match for the one you saved. DNA storage is a proven science, but it faces 4 hurdles before it becomes the industry standard. These are not failures, but the final steps in a technological revolution. 1. Throughput. We must continue to increase the speed of writing and reading to handle global data flows. 2. Total cost of ownership. While DNA is cheap to maintain, the initial printing cost must fall. However, as it scales, DNA will obliterate the need for massive, power-hungry data centers. 3. Security. Every system must be biosecure, ensuring that data sequences are never confused with harmful biological organisms. And 4. Standardization. We need universal rules to ensure a library built today is still readable in the distant future. Solving these challenges will transform the competitive landscape. DNA is moving from a mystery of the natural world into the permanent backbone of human memory. We are finally building a library that will never burn and a history that will never be forgotten.